Well, good morning, good afternoon, or good evening, ladies and gentlemen. Thank you for joining us for another installment of our video PowerPoint series for Unit 7, Mendelian Genetics. So today, what we're going to be talking about are primarily dominant and recessive traits, as well as the history of Mendelian genetics. Basically, the steps that we took towards what we now know as modern genetics. So we're going to be talking about Punnett squares, dominant, recessive, and how all of these factor into determining, well, what makes you you. So without further ado, everybody, I'll see you all in the next slide. Now, just in a disclaimer statement here, due to the fact that there's a lot of info on these slides, there will not be any video of me in the upper right hand corner this time, so you're just going to have to go ahead, sit back, and listen to the soothing sounds of my voice. Well, let's go ahead and get started. So the first thing that we need to discuss here is a man by the name of Gregor Mendel and his hereditary genetics. So, Gregor Mendel was a scientist and also a clergyman, and what he worked with primarily were garden peas. The reason why he chose these was because they are self-pollinating, so they can go ahead and self-fertilize. Also, they were very easy to go ahead and cross-pollinate, so very easy to cross from one to the other. And they had very distinct and clear traits. For example, they came in either pure white or pure purple. So as you can see here, we got a pure purple pea plant and a pure white pea plant. And these are what we call traits. So a trait is any single feature that an organism has. So how tall you are, your eye color, hair color, whether or not you have dimples, etc. So what Mendel discovered here was he took the parent generation, or the pea generation. So in this case, we've got two parents. We've got one tall and one short. So he went ahead and crossed these two plants. And what he noticed was in the F1 generation, or the first generation of offspring, he noticed that they were all tall. So he crossed a tall with a short and got talls primarily. So then he went ahead and crossed two plants from the F1 generation, or the first generation. And sure enough, in the F2 generation, so I guess if you wanted to compare this to, family, the P generation would be the parents, the F1 would be the kids, and the F2 would be the grandkids. So in the F2 generation, what he noticed here was we had primarily all tall, but we also had one short. So we had three tall plants and one short. Whoa, wait a minute here. Something's going on. And Mendel took note of this. So, just in case you didn't catch that, we have a short plant. So, where do you think that short plant came from? I mean, we crossed talls and we got a short. Was it hiding? Was it just kind of hanging out? Was it sitting back waiting for everything to go ahead and stop and then was like, hey, check it out, I'm a short dude? No. So, these are what we call recessive traits. Recessive traits primarily hide behind dominant traits. So, sexual organisms have two of each trait. If you think back, these are what we call homologous pairs, or variations for a trait. One of each pair is passed on to the offspring by each parent. You get one from your mother and one from your father. So in this case, the different forms of the same trait are what we call alleles. And we have two different alleles here for height. So the trait that we're studying in this experiment, if you go ahead and take a look, would be the height of the plants. Now the alleles, or the two variations for that trait, are what we would have are tall and short. The two different alleles, or variations for the trait, plant height. Now other traits that Mendel looked at were things such as height, tall or short, flower color, purple or white, flower position, axial or terminal, and he also looked at the peas themselves. So the seeds were either yellow or green, round or wrinkled, and the pod color was either green or yellow, or smooth or constricted. So which of these words below is a synonym for allele? Would it be trait or variance? Or maybe they're going for the odd answer, and maybe it's seed shape. Who knows? Well, if you said alleles, then congratulations, you would be right. Alleles are nothing more than variations for a trait. So, let's go ahead and look back at that first example, shall we? So our parent generation, we crossed one tall and one short. What trait were we looking at? And based off what we're seeing, considering the first generation we got all tall, what do you think the dominant allele would be? And if you think that's the dominant, which one do you think is the recessive? So if we take a look at this, we'll notice that we crossed tall with short in the P generation, and we got all tall offspring. So the dominant trait here must be tall. Meanwhile, in the second generation, the short showed back up. So in this case, if it was hidden during the F1 generation, we can assume that short would be the recessive trait. Now, if you take a look at the percentages here, in our P generation, or our parent generation, we've got a 50-50 ratio, one tall to one short. In the F1 generation, if you count them up, we got 4 tall. So we have 100% tall and 0% short. Meanwhile, in the F2 generation, we've got 1, 2, 3, 4, 5, 6 tall and 2 short. So we have roughly 75% tall and 25% short. But the real question here is, why? I mean, why does this happen? Well, stay tuned because we're going to go ahead and find out. So, Mendel proposed that factors segregate or separate from pairs to be passed on to the offspring, one from each parent. Does this sound familiar at all? Think back to, I don't know, mitosis and meiosis. Think the old PMAT. So, if you go ahead and take a look at this, what are we looking at? Now that is some high quality animation right there. So homologous chromosomes split up during anaphase 1 of meiosis, so that only one allele for each trait gets passed on in your gametes, or the sex cells, the sperm and the egg. So with the help of anaphase 1, our homologous chromosomes separate so that each cell has a specific variation that will be passed on to the offspring. So the pair of alleles you have for any trait is what we call your genotype. Now let's say, for example, we're looking at the shape of your nose. Let's pretend that round nose is dominant to pointed nose. Now usually the letters are used to represent alleles are uppercase for dominant and lowercase for recessive. So let's go ahead and assume that since we're working with a nose here, let's just use the letter N. So capital would be dominant, right? So we'd have an uppercase N for dominant and a lowercase n for recessive. So the genotypes are the potential combinations of letters or alleles that we could get for the offspring. So let's think about this for a sec. Now, what type of genotypes or combinations, of two by the way, could we get for big N and little n? Well, we could have big N, big N, big N, little n, or if we can have big N, we also need to have little n, little n. So we have three different genotypes or variations for nose shape. Big N will be recognized as the homozygous dominant, or two dominant traits. The big N, little n, or the combination, would be recognized as heterozygous, or the hybrid. Meanwhile, little n will be recognized as the homozygous recessive, or two recessive alleles. So the pair of alleles you have for any trait is called your genotype. So to really go ahead and drive this home, big N, this genotype could be described as homozygous dominant, or purebred, in this case, round. For the middle, the genotype big N, little n could be described as heterozygous, a hybrid, or carrier, because we've got one of each. And finally, the genotype that can be described as little n would be homozygous or purebred recessive. Two alleles for the recessive trait, in this case pointed. So the only way we're going to get a pointed nose is if we have two little ends together. So how did you get your genotype? Now each set of alleles is a homologous pair, one from mom and one from dad. So let's say you received a big N from your mother and a large N from your dad. What genotype would you have? So big N, would you have a rounded or pointed nose? Remember, rounded is dominant and pointed is recessive. This would give you a homozygous dominant pairing, giving you a round nose. Now, round nose in this case is what we would call your phenotype, or the expressed feature you can see. So the genotype is the letter combination, whereas the phenotype is the condition that it represents, or the physical characteristic. So what do you think this person's genotype is? Big N, little n. That would be heterozygous. So the phenotype, would it be rounded or pointed? In this case, it would be round, because we still have that large N. And remember, round is dominant to pointy. So as long as there's a dominant letter there, it's going to show the dominant phenotype. Remember, always the dominant overshadows recessive. So big N, round, will always overshadow pointed. Now in this case, we have little n, little n. What do you think this person's genotype is? We would call that homozygous recessive. Now considering we have two of the recessive traits, or the recessive alleles, this person's phenotype would be pointy. So the recessive alleles are only going to show if no dominant is present. So the only way you can show the recessive phenotype is if you have two of the recessive alleles, in this case, two of the lower cases. So Gregor Mendel's research and principles. Now there's quite a few here. There's a short video, so if you wanted to go ahead and click the link, I know it's kind of hard to see, but that'll point you in the right direction for a little history on Mendel. Now Mendel had a couple of principles. The first was the principle of segregation, which states that every individual possesses a pair of alleles for any particular trait, and that each parent passes a randomly selected copy of one of these to its offspring. The offspring then receives its own pair of alleles for that trait. Now factors for a particular trait occur in pairs. So basically what this is saying here is the pairs will split and each parent passes one randomly selected copy onto the offspring. The second is the principle of independent assortment, which states that genes for different traits can segregate independently during the formation of gametes. So factors segregate randomly. What we're talking about here is that during anaphase 1 of meiosis, primarily your homologous pairs are going to separate independently from one another. Sorry, I was talking too much. I'm starting a little dry mouth here. Nothing a little T can't solve. And finally, we have the principle of dominance, which states that alleles are dominant and others are recessive, and the dominant factors will mask the recessive ones. So the dominant trait will always show over the recessive. So let's take a look at our example here. What trait are we looking at? We've got two different colored flowers. So in this case, the trait that we're looking at is flower color. What are the alleles in this case? Remember, the alleles would be our variants for the trait. In this case, our variants would have to be purple or white. Which one seems to be dominant? Well, if you look closely here, when we cross the two of them, the offspring was purple. So we can assume, considering that it's got the genotype big P, that purple is dominant, whereas white is recessive. Now remember, only one of each allele is passed on from each parent. So, let's go ahead and take a look at mom here. What is mom's genotype? Remember, genotype is the combination of alleles or letters. This would be heterozygous. So heterozygous because we have a combination of each. So if it's big P, little p, is that going to be purple or white? Remember, purple is dominant, white is recessive, so this would be a purple flower. Now with Dad, on the other hand, we also have big P, little p. So Dad's genotype is also heterozygous. So if we cross the two of these together using a Punnett square, this is what we're going to do. So if this egg meets this sperm, all you're doing is just going ahead and combining the letters, kind of like a grid. So we would take the dominant P from the mother, the dominant P from the father, and this option would be big P, which would result in a purple flower. Now what if we had this one over here, with a little p from the mother and a big P from the father? This would give us big P, little p, or a heterozygous purple flower. Now what if we took the big P from the mother and the little p from the father? Once again, that would give us a heterozygous purple flower. And finally, if we took the two recessive white traits and we went ahead and crossed them, that would give us a homozygous recessive white flower, little p. This is what we call a Punnett square. It's a real nice tool that goes ahead and allows us to predict the probabilities about offspring, as long as we know the variance in traits for the mother and the father. Now we'll be using these a lot here throughout the unit, so you make sure that you'll have plenty of practice as we go through. So if you don't quite get it yet, that's okay. Follow along, we got plenty of time to go ahead and get you caught up. So let's go ahead and move on and see where this takes us. So what we can do here is use these percentages to predict the offspring. So how many purple flowers are expected? There's a 75% chance, or 3 out of 4, that the offspring will be purple. Whereas there's only a 25% chance, a 1 out of 4 chance, that the flower will be white. So how many homozygous dominant options do we have? Now remember, homozygous dominant would be 2 dominant alleles. So we have a 25% chance that it will end up as big P. For heterozygous, there's about a 50% chance, so 2 out of 4, that the offspring will be heterozygous, or having one trait from each. And finally, for homozygous recessive, there's only a 25% chance that we'll have the little p, resulting in the white flower. So, let's say that we're looking at a specific disease, the A disease. So the healthy allele, meaning no disease, is capital, whereas the disease allele would be lowercase, little a. So what is the probability that this couple will have a baby with the lowercase a disease? Now remember, since it's recessive, the only way that they can have it is if they have 2 lowercase a alleles. So considering that the mother and the father are both carriers for this, they're both heterozygous, if we did a Punnett square, you would notice that we would have a 25% chance of it being big A, a 50% chance of it being big A, little a, or carriers, and a 25% chance that the offspring would have the disease. Now, what's the probability that this couple will have a baby who is a carrier for the disease? Now, same thing, think back to what we just did. A carrier would have the same genotype as the parents. So one of each. One healthy allele and one disease allele. So a carrier is known as a heterozygote. So they won't show the disease, but they can pass it on to the offspring. In this case, we'd have a 50% chance, or 2 out of 4, that the offspring would be a carrier. Now, once again, being a carrier means they don't have the disease, they're not going to show it, but they could potentially pass it on to their offspring. Now, let's go ahead and think back to the A disease once again. What if a homozygous dominant man marries a carrier woman? So if it's homozygous dominant, remember we're still looking at the A disease, so big A is healthy, and little a has the disease. So in this case, if dad is a homozygous dominant, now dominant being healthy, he would be big A, big A. Now if mom's a carrier, that means that she doesn't quite have the disease, but she could pass it on. And she is heterozygous for this. So that means that she would be big A, little a. So let's go ahead and set up our Punnett square. In our first box, if we go ahead and look down here, we'd be crossing the big A from the mother and the big A from the father, giving us the combination big A, big A. In our next box, if we cross the little a from the mother and the big A from the father, we would have big A, little a. In our third box, big A once again. And in our fourth, we would have big A, little a. So our offspring genotypes here, for heterozygous dominant big A, there would be a 50% chance. As you can see here, we have a 50% shot that we'd have big A. Heterozygous would be big A, little a. And sure enough, there's a 50% chance that that could occur. And there's a 0% chance that the offspring would actually have the disease or be homozygous recessive. Now those are the genotypes, which remember the genotypes are nothing more than the combination of alleles or letters. Now let's go ahead and look at the physical characteristics of the phenotype. So remember, healthy, we're including healthy as carriers. Because technically they are healthy, they don't show the disease, but they could pass it on. So the potential here that we would have a healthy offspring is 100%. There are no little a's here. So we would have a 0% chance of having a diseased child. Now let's do this one more time. Let's say a carrier man marries a woman with the disease. So carrier man, if he's a carrier, that means he's heterozygous. So he'd be big A, little a. And if the woman has the disease, that makes her small a, small a. So we set up our Punnett square. Let's run through. We have our first one. We'd have big A, little a. We have our second, which would be big A, big A, little a. We have our third, which would be little a, and we have our fourth, which would also be little a. So if we go ahead and do the genotypes here, for homozygous dominant, that's a 0% chance there's no big A. If we're looking at heterozygous, in this case, the upper and lower case, there's a 50% shot. Whereas if we look at the homozygous recessive, little a, there's a 50% chance. So, the percentage of healthy versus diseased would be 50% healthy, 50% diseased. And of the healthy, of the healthy, there would be about 100% of them that would be carriers, because they're big A, little a. So if we think about what we know now, what we have here in the first generation, if you go ahead and look at the percentages, we've got 50-50 for tall and short in the first generation. In the second generation, well, let's go ahead and say that big T here is tall. And let's say that little t is our short. So in our first offspring generation here, we got all tall, but since we crossed it with a purebred short, we know that this would have to be big T, little t for all of them. So that fits our description, 100% tall, but the genotype would be big T, little t, meaning they're all hybrids. Now if we cross two hybrids, remember there's a 75% chance you'll have the dominant trait and a 25% chance that you will have the recessive. So now we know why all this works. It's all about the Punnett squares, it's all about the probabilities, everyone. Well, surprise, guess you get to see me after all. Well, Ladies and gentlemen, that just about goes ahead and closes things up for today, so I'd like to thank you again for joining us for our first video PowerPoint here for Unit 7, Genetics and Heredity. So in summation, what we ended up talking about today was the history of Mendelian genetics, and we also talked about the various options for genotypes and phenotypes. Genotypes being the combination of alleles, or variations for a trait, and phenotype being the physical characteristic they represent. So for the genotypes, we talked about 3 different conditions you could have. We talked about homozygous dominant trait being the capital letters, so homozygous dominant would be 2 capitals. We talked about the heterozygous, or the hybrid, meaning that we had 1 large and 1 small, so we had a dominant and a recessive, and we finally talked about the homozygous recessive, which would be 2 lowercase alleles, or 2 recessive alleles. So 2 lowercase letters. So, to go ahead and finish this off, I'd like to thank you all for joining me today, and until next time, I will see you in the next video. You all keep it classy and stay warm out there, because, well, we're still in the middle of a snow day, so, hope you're enjoying it. Take it easy, everybody.